Hi, my name is Kristen Grosclose, President of the Smith-Kingsmore Syndrome Foundation. We are so fortunate to be partnering with Dr. Andrew Liu from the University of Florida College of Medicine, where he is studying mTOR and the circadian clock, which directly supports the SKS community. The foundation has chosen to support his research by raising $50,000 to fund a postdoctoral researcher to help Dr. Liu create a personalized treatment plans for each known SKS variant. Please take a moment to listen to Dr. Liu and hear how your donations are directly supporting his research and how you can help improve the quality of life for SKS families.
Well, thank you, Dan, for the opportunity. I'm honored to be part of this society. I got my PhD in biochemistry from University of Michigan. I went on to get training at the postdoctoral level on molecular genetics at Scripps Research Institute in La Jolla, California. My lab studies, by the way, I'm from the University of Florida College of Medicine. My lab studies, let's see if I can say it, molecular, cellular, physiological mechanisms of the circadian rhythms in mammals. We use mice and cultured mammalian cells as model systems to study. So circadian rhythms, I'm sure all of us are familiar with circadian rhythms. At the central level, our day and night are distinctly different. You know, our number one is the sleep-wake cycle, and associated with that cycle is the rest activity cycle, the feeding fasting cycle. That also includes our body temperature, our heart rate, and our blood pressure. All of these are different day versus night. And abnormal rhythms are not good and actually an indicator of symptoms or disease states, if you will. And these rhythms, that also includes the hormones, the neurological functions and hormones that are distinctly different. For example, cortisol, melatonin, and all of these rhythms originate at the cellular level in individual cells. So these are called circadian clocks at the molecular level in individual cells. So we can look at the clock we can think of it as a smartphone. You know, we have a lot of apps. The clock regulates these apps, right? I think the central question in my lab studies is what regulates the clock. Thinking again, the smartphone, when you travel from one place to another, your app actually can sense where you are and tell you the local restaurants instead of the restaurant in the original place. So in a way, the clock can sense the environment locally in individual cells, individual organs, as well as at the body level, where, when you travel from place to another. So my lab studies identify genes, pathways, and physiological, pathological states that regulate the clock, which then regulates physiological processes. So these signals that regulate the clock can be synchronous. For example, if we conform to the sleep-wake cycle, the light-darker cycle, that's good. But if we reverse that cycle and the clock can tell, well, that's not right. And so the pathways we're studying include the energy sensing, the metabolic and energy sensing, oxidative stress. In recent years, we have been studying inflammation. So all these different physiological, pathological states can affect the clock function. At the genetic level, the gene that the reason that we got connected is mTOR. A few years ago, we discovered mTOR regulates the clock. So whether it's genetic change or environmental change that could change the mTOR activity can change the clock function, and therefore it can change the sleep-wake cycle, and all other associated physiological processes can change. So I think this is the background of what my lab studies, and a little bit link there, why we are studying mTOR. I had the honor and opportunity to visit Cincinnati when we had the first symposium, and I see parents and the kids over there. I also look at every single mutations that that have been discovered so far. What I can tell is all the mutations are not the same. All the patients are not the same. I think overall, ultimately we can tell their responses to nutrition, responses to rapamycin treatment are likely not going to be the same. So for treatment, what I have been trying to say is we need to consider precision and personalized medicine. And that is to be able to optimize and refine the treatment strategies. But that refinement is based on science, based on the studies about these different mutations, different activities, and their responses to drug dose and diet, to try to, this is the key word, to try to achieve the optimal mTOR activity. And because mTOR is so important, it's the master intracellular sensor for the metabolic and energy states, which then regulates a lot of things, including the clock function, including the sleep-wake cycle. So our hope is to be able to understand these mutations and optimize the mTOR activity to the level that is appropriate, or if you will, optimal. That not only improves the metabolic states and lifestyle, but also minimize side effects, right? So especially in this type of treatment that is long-term, right? Long-term in young kids. So that's my mission, and that is also my passion. That is to be able to study these mutations. Hopefully, our results can offer some insights into the treatments in a precise and more personalized manner.